you know what? Hey, why don't I record this lecture? This is great. Hey, so for whoever can't make it, we're going to have a recording. Good. So welcome, everyone. This is 6047/6878/HSD507. And it is the first lecture of the term. So we're going to be talking about computational biology, and specifically genomes, networks, evolution, and health. So my name is Manolis Kellis. I'm a member of the MIT Electrical Engineering Computer Science Department. My lab sits at CSAIL, just 5 floors above you. So we're going to be having a lot of meetings over in the shared space on the 5th floor of the Dreyfus Tower. And I'm also a member of the Broad Institute. So what are we going to be talking about? We're going to be talking about phylogenetics, about epigenomics, about gene expression analysis, regulatory motifs, regulatory networks, human population and migrations and evolution, and personal genomics and disease. So the TA, who is so bravely taking care of setting this up from thousands of miles away, is Koi Nguyen. So she's actually a PhD student in the Department of Biological Engineering, and she works on chromatin biology, genomics. My own research is on comparative genomics, gene regulation, evolution, genomics, phylogenomics, genomics, and pretty much a lot of the topics that we're going to be talking about today. So first, I'm going to be talking to you about some administrative issues surrounding the course, and then I'm going to be telling you a little bit about sort of how the course is organized and what's the content, and sort of what do we do to actually give you a grade. So this is the staff, so myself and Koi. We're going to have a lot of other help from members of my lab who are going to be helping mentoring a lot of the projects. And then this is who you are. So this is for the subset of people who were pre-registered and actually had time to fill out the survey. So you can see that the course is mostly taken by undergrads in their late years, and then grads in their early years. Basically U3, U4, and then G1, G2. Hello? Is that a question? I can hear somebody speaking. Can you speak up? I can't hear you though.
No one in the class.
Oh, strange. Oh yeah, so I guess Laura, you must be the one. So I'm gonna mute you. There you go, I muted Laura. All right, and in terms of courses, so basically 6-7 is a lot of the U3 and U4 who are taking the course, and then 6 is a lot of the G1 and G2, and also some 7, so about 10% of the students are actually coming from biology. And then quite a breadth of topics for more than a quarter of the class, which is very exciting. So you should also not be intimidated. About 40% of you said that you are currently doing research in computational biology. This is actually a very good number. It basically says that you're going to have a lot of experts that you're going to be able to interact with. And these are the specific topics that people are currently working on. So this will be part of the slides that will be updated on the web after this lecture finishes. So people from, you know, many different labs, but this is only 40% of the students. The other, let's see, so welcome, Kaitaf Cheat. The other 60% of the students have actually never done research in computational biology before. So this is meant to be an introductory course, and that's what we're going to be doing. We're going to be both introducing the topic and also touching on frontiers of the topic. I do not expect you to be able to read this. But basically, these are all of the answers as to why are you taking this class and what do you hope to learn from it. And I'm going to just read some highlights. So, by the way, can anybody actually read this? I don't expect you to. So, Koi, have you double-clicked to maximize the presentation?
Let's try. Yes, no, it's full screen.
All right, perfect. Good.
Yeah.
Can you actually see me as well, or you can only see the slides?
You can also see your face. Yeah, in a little pop-up.
Say it again. Oh, in a little pop-up. Great, yeah. Perfect. Can you move the pop-up to the side so that it doesn't hide any of the slides?
Yeah, I think it's okay.
Okay, beautiful. All right. So basically, why are people taking the class? So there's a lot of classes of responses. So some of them are actually strong in biology and they want to learn more about computation. Others are, most of you guys are strong in computation, want to learn about biology. And about a third or two-thirds or half of the people are actually currently studying both computer science and biology and want to learn more about both. So, cool. So, and then, you know, you can see all the, basically, some of them are saying, you know, I want to learn about current research frontiers. I'm debating about career options. I'm debating about, you know, PhD projects and so forth. I should have turned off the sound when you joined. Then I actually asked everybody about their interest in specific topics for the class. All right. So, what's going on?
So when people join the meeting, it has a big doorbell sound.
Yeah, I know. So let's see.
That I cannot stop.
Let's see. Just 1 second. There's a setting for that.
Okay, I don't know where it is.
It's somewhere. Let's see. Well, this will hopefully end as people stop joining.
Okay.
All right. So then the other question on the survey was basically how much are people interested in various topics in the class? And it was actually quite reassuring that almost no one responded none for each of the different topics. And then there was very few topics where there was low interest, but those topics also had a lot of medium and high interest. So it's quite reassuring that basically we can, you know, continue going. I actually considered changing the course dramatically based on the responses, and they kept trickling in, and I was quite reassured that there's a lot of good interest for nearly all of the topics. And then this is other topics of interest that people suggested. So biological images, there were a lot of suggestions for structural biology, and so forth. So unfortunately, we're not going to be touching upon protein structure. You know, this course is specifically focused on genomics, not as much proteomics. But there was a very good suggestion here on data visualization and presentation. So can I see a little bit of a chance for who would be interested in data visualization and presentation as a topic? Could we add a topic on data visualization and presentation?
That looks like maybe 10 people. 10, 15?
Say it again?
That looks like about 10 to 15 people. If you
10 to 15 people. Good. So basically maybe 40%. Is that it?
Yeah.
Okay, cool. So yeah, we may do something like that. We might do it in a recitation, specifically as we're getting closer to the final project. Should have turned off this earlier. So this is actually one of the settings in Zoom. So I don't know if I should bother doing that now, but let me try to, yeah, I'm just not gonna bother doing that now, sorry. Yeah, I shouldn't do that now. I think it has kind of stopped, so yeah, sorry for all the beeping. Okay, so basically, yeah, we probably will do and others such as deep learning, network biology, and so on and so forth are already covered. Network topology, you know, this was my favorite, everything. So yeah, this is actually one of the downsides of the course. The most negative comments we get every year is that the course covers just too much. And I'm relieved to see everything here because we kind of err on the side of everything rather than nothing. All right, so here's the course information. So we're usually going to be physically meeting in 32141. Recitations are scheduled by the registrar, and I believe they're on Fridays at 3 p.m. in 4237. We're going to update that if it's correct. And then all you need to do to go to the course webpage is go to compbio.mit.edu/6047. And that will redirect you to the Stellar webpage where you will see all of the materials. So on Google, you can actually add a public calendar called 6047 lectures, and that will tell you the lectures for each class. And here's our goals for the term. Our goals on one hand are to introduce computational biology, and on the other hand is the ability to tackle research questions. So basically, we're both going to be dealing with fundamental problems in computational biology, looking at the algorithmic and machine learning techniques that we use for data analysis. and research directions for active participation in the field and understanding how the methods work. And it was actually kind of nice that, when people said, why are you taking this class? They said, I've always wanted to know how these particular tools that I'm already using, how they're actually working. In fact, that's going to be a lot of what we do in the course, basically understanding not just how to use different tools, but how the tools actually work in order to then understand how to even improve upon them. And then a major component is the ability to tackle research. So there's going to be problem set questions that teach you about algorithmic rigorous thinking, a lot of programming assignments that are hands-on, giving you experience with real data sets, and then a final project experience, which is actually term long, where you propose and carry out your own original research, and then you present the findings in a conference format, both written and oral. So let's talk about the content. So there's two dualities in the course. The first duality is about computation and biology. The second duality is about foundations and frontiers. On the first one, we're going to be both learning about important and relevant and current biology, so very important biological problems. And we're also going to be learning about fundamental computer science, so general techniques and principles. So nearly every module and nearly every lecture will cover both computational tools that you can use in many other places and biological problems that people care a lot about. The other duality, on the y-axis if you wish, is foundations and frontiers. So foundations, we're going to be both looking at well-defined problems, general methodologies, the classics of the field, and that's going to be usually at the start of each module. And then we're going to be looking at frontiers in the last part of each module, looking at in-depth look at complex current problems, open questions, where we combine a lot of the techniques that we've learned in the foundations across the term, and then apply them to sometimes open questions or very recently published work. And that will open up the projects and research directions. So every module will basically correspond to an active area of research. We're going to be talking about comparative genomics, about genes and transcripts, about gene regulation, epigenomics, regulatory genomics, about population variation, looking at genetic variation, human history, heritability, and looking at evolution, phylogeny, evolutionary signatures, whole genome duplication, assembly. And then in frontiers, we're going to be looking at both personal genomics and disease, as well as 3D genomes, and so on and so forth. And for every module, the first half will be foundations, and that will include a lot of computational tools that are very broadly applicable, like dynamic programming, string matching, hashing, hidden Markov models, expectation maximization, Gibbs sampling, clustering, classification, feature selection, support vector machines, context-free grammars, phylogenetics, and so on and so forth. And then for each module, the second half will be very much the frontiers as to areas that are still being developed. Every year there's new material that is making to the class. So this is the basic organization. So here are the different modules. So module 1, aligning and modeling genomes. Module 2, gene expression for genomics. Module 3, regulatory genomics and networks. Module 4, population genetics and disease genomics. Module 5, comparative genomics and evolution. Then the quiz, and then module 6, a lot of current research directions, including single-cell genomics, human phenotypes and PHEWAS, so electronic health records, cancer genomics, CRISPR, genome engineering, and so on and so forth. And for each of them, you can see the foundations and the frontiers, and again, foundation frontiers, and the chapters corresponding to each of them are here. Okay. So this gives you an idea of what the term will be about. It's quite a dense chart, but it basically tells you for every week, what are the different dates, what are the different modules, and so on and so forth. It also tells you about the problem sets. So basically, every problem set is going to be handed out at the beginning of each module, and it's going to be due at the end of each module. Basically, the Monday evening or the Tuesday right before the next module starts. So PS1 will be out today, and then due next Monday, in 3 weeks actually, at like midnight, and then PS2 similarly, PS3, PS4, PS5. And at the same time, there's this column here about the project. So basically, along with the PSET, we're going to be building up your project proposals, and first constructing your own profiles that we're going to be sharing with the rest of the class in order to help you find teammates. And then learning about what project areas every team is interested in, submitting a proposal by the end of the third PSET, that basically, tells everybody, hey, here's what we're going to do, and we're going to have actually class presentations where we're going to be going over these proposals. These are actually going to be the green parts that are interspersed. You can see that at the end of each module, there's a meeting where we get together for mentoring. So basically all the green parts are the mentorship. And that's when we're going to be getting together in the 5th floor of the Dreyfus Tower, along with members of my lab and other friends and collaborators, to basically help mentor these projects. So basically, first, we're going to be helping you find teams and match up with mentors. Then we're going to be helping you plan your projects and then giving you feedback on these projects. And then after you submit your project proposals, we're actually going to have an NIH-style review panel, which will basically give you feedback as if your proposal was a grant proposal. So this is something that you don't get too much training doing, so we thought it would be a good idea to do it in the context of a class, to basically both provide feedback to your peers and to also receive feedback from your peers. And the beauty of it, and something that a lot of people have described through the years in the class, is that even as they're writing critiques for other students' projects, they think critically about their own project at the same time. And then they say, how would I view my own project critically? What are the points of failure? What are things that I should be more aware of? Maybe I should, try to address them early. Maybe I'm relying on a data set that will not become available, and so on and so forth. So that will basically be discussed in a group. There's going to be 3 or 4 reviewers for every project. And then you're going to receive back feedback, and you're going to have 2 weeks to respond to that feedback, and then submit a revised proposal. That sometimes will say, the feedback was great, I don't need to change that much. And other times we'll say, well, I realize this whole project won't work. Or hopefully most often it will say, well, here's a few ideas about how I can change the course slightly to make the project work. And a few weeks after that, we're going to have a mid-course report that is basically helping you get to the final report in time and with good progress. Don't panic if you don't have most of the results there. But what you should have is at least an outline of what you're going to be creating by the end of the term, so that between the mid-course report and the final report, you're mostly filling in blanks rather than still deciding on a project and still exploring and so on and so forth. So you're going to have 2 weeks after that to really bring things together. So there's a lot of hand-holding. There's a lot of sort of forcing you to do things on time, and talking with your mentor, bouncing ideas back and forth. And our goal is to really help you achieve something great before the end of the term. And this has been true year after year. I've been consistently blown away by the quality of the final projects that people are able to come up with. I'll take a pause here and see if there's any questions about the general structure of the class, and see who's excited or who's panicked, or maybe both. So Koi, any feedback, any hands up, or any questions?
Any questions? Yes.
Yeah, I have a question about the project. Like, could that be like an extension of like the current like lab computational project that, like, if that's the case, like, are there any like potential like conflict of interest issue or like anything like that?
Manolis, did you hear that?
Yeah, I could hear that. So it's a fantastic question. It's a question we get every year. As you can see, 40% of you are already working on a project. We would like you to define a component of that project that is novel and that is very well related to the class. Basically, if you can say, listen, here's what I'm doing already, and here's the new thing that I'll do for the class, even if in the end it ends up contributing to your publication or to your thesis, that's perfectly fine. But what we'd like to do is really have it start in the class and have it be very relevant to the material shown in the class. Does that make sense? And then the other thing we'd like you to do is, especially if you're already working on a project, find a collaborator, find another student who's not already in your research group that you can collaborate with. Basically, we really encourage people to do team projects, and 3 is too many, 1 is too few. We really want people to do projects in teams of 2. The reason we don't like 3 is because a third wheel is always, bouncy. With 2, everybody's carrying their own weight. and you know, the management of the team is much better. If you make a strong justification for 3 and you have a very clear division of labor, then, you know, we will of course accept 3. And similarly, if you make a very strong justification for 1, we will also accept 1. But basically, the justification should not be, hey, I'm already working on this, so I just want to do this solo. You should really team up with somebody with complementary expertise, if possible, and then do something that you wouldn't have done if you weren't taking the class. Any other questions? All right. So I'll carry on. So basically, in terms of textbook, we're going to be using a lot of class notes. So here's 3 recommended textbooks. They're not required, but if you want to learn more about the different topics covered, we recommend Biological Sequence Analysis by Durbin et al. Introduction to Bioinformatics Algorithms by Jones and Pesner, and then Pattern Classification by Dada, Hart, and Stork. So these are all available, you know, at the BU Coop, at the MIT Coop, on Amazon, and they're also in reserve, but they're not required. So it's just good general reading to broaden your horizons. The most important resource will be this course notes that have been assembled by students like you over the course of the years. So I will update this with the lectures from last year, which are already posted, and I just didn't update this. But you will see it's about 500 pages worth of notes, and it allows you to go into depth about each of the lectures. So I really recommend you browse through it for the topics that you're the most interested in, and you will be all choosing a lecture to Scribe. In fact, I saw several of you signing up to Scribe different lectures as the course was starting, as the lecture was starting. So I saw many of you on the website. For the topics that you're the most interested in, it really helps to number 1, read the notes, and number 2, improve on the notes. So sometimes you feel that you really grasp something well, but trying to explain it for others really goes much more into depth. So again, this is being compiled by students like you, including you, and then every lecture will have a dedicated scribe who will take notes on the lecture, and then this will build on notes from previous years, and you will complete the draft of the scribe notes before the previous lecture, and the final draft will be due about 6 days after the lecture. So some lectures will need more work, specifically later in the term, and then we would like to have multiple scribes. And then some tasks might be better suited to you rather than just scribing. For example, if you're very comfortable with graphics and you know, illustrations, you're welcome to join just you know, task force on figures. Or you know, if you're an expert in LaTeX, you can help with references and layout and macros. Just let us know if you'd like to scribe for something beyond just a single lecture. And then you can do that by simply adding a line to the existing Scribe sign-up list, and then you'll see it there. So there's a Dropbox called 6047 Book, which contains basically all of these different directories, and there's a make file for each of them, and images for each of them, and then a text file for each of them. So you're going to be editing this text file, and then running this make file, and then compiling these images here, and then that will be creating you know, a PDF for every one of those, and then a compilation for the whole class. So if you haven't signed up already, here's the URL, QJRMGG in this particular uppercase, lowercase. So basically do sign up. So these are the different lectures and different topics, and then you can sign up now. We're going to be posting, gosh, I think I should be doing this recording every lecture. So maybe I'm going to start creating a Zoom link for every lecture, and maybe recording it that way. And that will give you, you know, the ability to go back and review any one topic. We're also going to have, actually we already have a form where you can enter your feedback for every lecture. That basically tells you what was your interest in the topic before you heard the lecture. What was your interest in the material that was actually presented, not just the overall topic, but the material within that topic. And then, you know, the quality of the slides, the explanations, the usefulness of the lecture notes, and whether the questions were answered adequately. And then the pace, basically how difficult was the material, how much material was covered, how was the pace of the lecture, and lastly, how much do you feel you comprehend each topic, between 20%, 40%, 60%, and so on and so forth. So you don't have to do this for every lecture, but if you do, it will be very helpful, and it will allow me to know, how things are moving. Again, we tend to err on the side of covering too much. If you give us feedback early, we can adjust that and cover less. In terms of homeworks and quizzes, so there will be 4 problems in every problem set, with every problem emphasizing 1 lecture or 2. And then there will be a both practical problems and theoretical problems. The practical problems will give you experience in techniques, writing code, downloading data sets, carrying out analysis, interpreting the results, and then learning about the behavior of some problem or some method. And then the theoretical problem will be mostly a pen and paper problem, exploring algorithmic, statistical, machine learning aspects in detail. And then there will typically be a 5th problem, which will be an advanced problem for students taking 6, 8, 7, 8. These will be due probably Mondays at midnight, but our late policy will be that we're flexible. And if you're more than a few hours late, let us know in advance. But if you are just a couple of hours late, it's not the end of the world. We're not here to penalize you. We're here to help you learn. And you'll submit all the homework online through the Stellar page. We won't distribute solutions. You'll know if you've solved them, and we'll know if you've solved them as well. We're going to have an in-class quiz. This will come after the 5 modules and before the last module. So it's not a midterm. It's not a final exam. It's a quiz. It's supposed to be friendly, fun, interesting, cute, and fuzzy. The goal is to demonstrate mastery of the material in the first few modules, and then to understand the key points emphasized in the lectures, and to understand the subtleties that were revealed in the P-sets, and to apply them to solve practical problems. So we will have about 15 true-false questions, then maybe 8 deeper understanding questions, and then 2 or 3 practical problems, and then maybe 1 design problem, where you either provide short answers or work through a simple algorithm or actually modify an existing algorithm. It basically demonstrates both knowledge of the topic and the ability to come up with a new idea. So the final project is actually a whole term project, so I should be calling it term project instead. The goal is to create original research in computational biology, to frame a biological problem computationally, gather relevant literature and data sets, solve this problem using new algorithms or machine learning techniques, and then interpret the results biologically. So you're starting with biology, gathering data, making it practical, and you're moving to the computational realm, solving it in that realm, and then coming back to the biological realm to interpret your results. And we will also, of course, be testing your ability to present your ideas and research. So you're going to be crafting a research proposal, which will be very helpful for fellowships and grants. You're going to be working in teams of complementary skill sets, reviewing peer proposals to find flaws and suggest improvements, which will again help your own critical thinking about your own projects. You will receive feedback, which is always constructive feedback, not just saying, hey, this sucks, but hey, this is great, here's how to make it better, about revising your proposal, and you will revise your proposal accordingly, which is extremely important in the real world, just incorporating feedback, something that you don't get to do very often in class projects. And then you're going to be writing up your results in a scientific paper format, and then presenting a research talk to a scientific audience, which will be your peers. And then the term project experience will again mirror the real world of sort of how this actually happens. So again, it's a team project. So this is actually your own photos. This is everyone who's registered in the class. So please make an effort to meet your peers. So talk to each other. We will have many mentoring sessions to actually meet each other. We will create profiles for every one of you and then share them with every one of you so that you're able to find people with complementary skills and interests. And the goal is very much to not just train you, quote unquote, but also to build connections to other students who will be your peers as you guys go to take over the world, and who will be your peers as you work on class projects, as you go to grad school. A lot of people who are very, very good friends in other institutions met actually through this class, sometimes worked on projects together, and that can actually help shape, your research directions and so on and so forth. So make an effort. All of you guys look super friendly. So meet each other, be social. It will pay off in the long term. We talked a little bit about the final project before, about creating your profiles, building teams, creating the proposals, reviewing these proposals, revising these proposals, creating a mid-course report, and then the final report, and lastly, your final presentations on the last day of class. So the goal of the milestones is to ensure that there's sufficient planning and feedback. For inspiration, we're going to be actually sharing projects from last year and recent papers. And then the goal of the proposal is to establish milestones, deliverables, and expectations. And the goal of the mid-course is to really see the endpoint, outline your report, finalize your methods and figures, and basically say, hey, figure 3 is not ready yet, but here's what I envision there. And figure 4, is not near yet, but here's what I'm envisioning. So the periodic mentoring sessions will have both senior students in the group and postdocs who serve as your mentors. We'll have group discussions to share ideas. I'll provide guidance to each of you personally and feedback. And you will be working with Koi and with other members of the group. And again, the peer review will allow you to both think critically about the peer proposals and receive feedback, respond to critiques, adjust your course, and so on and so forth. Something that we've only started doing recently is engaging the communication lab at MIT. This is a free resource for peer feedback from trained ECS grad students. So people go to the Com lab to work on their resume and their CV for grad school applications, faculty packages, oral presentations, fellowship applications, thesis work, manuscripts, and so on and so forth. So we are actually going to be giving you a 2-day extension for every one of your milestones if you have an appointment with the communications lab. So on the due date, if you tell us, hey, I've actually been to the communications lab and I'm incorporating the feedback that I got from them, you will automatically get a 2-day extension for every one of the assignments. Not for the problem sets, but every one of the project-related assignments. So this is hopefully good motivation for you guys to talk to them. I've had students and postdocs come to me and say, oh, I haven't talked to them because I write great. And I tell them, yes, you write very well, but you can always improve. And in fact, some of the people who are the most confident are very often making mistakes that are very, very common among trainees that you can overcome by speaking with the communication lab. So who thinks this is a good idea? Let me see a show of hands who's actually excited and interested about getting A, a 2-day extension, and B, talking to the Com lab. Awesome, good. So how many hands going?
I would say like at least 50%.
Awesome, good.
At least 50%.
Nice, nice. Great. So then lastly, finding a research mentor. So we will have, some guest lectures. We will have, we will invite students from other groups as well. And if there are topics that our own group is not currently working on, we have enough friends and peers at MIT and elsewhere that we can connect you with people working on these things, and they will then be invited to participate in the mentoring sessions and so on and so forth. So you will have a chance to meet your students and postdocs, and you can also bring your own collaborators and data sets from your own research experience. So again, the goal is to learn active research directions well beyond what's in the course material, and then it's a very living and breathing and changing field. So putting it all together, the grading will basically require 30% from the problem sets, 40% from the final project, 20% from the midterm, and then 10% from the scribing. 4 P sets, each of them about 7 to 10%, each of them covering 3 to 4 lectures. And then the final project is actually the big chunk with almost 40% of your grade. Again, mirroring this term-long experience and the many mentoring sessions and assignments that lead up to it. And I was again delighted to see so many people mention that they wanted to get research into the field as one of their motivations for taking the course. And then lastly, scribing will be 10%. So collaboration will be allowed, but you must work independently on every problem before discussing it. You must write solutions on your own, and you must also acknowledge your sources and collaborators, basically say, hey, we thought about this together, we worked together, in the end we wrote our own solutions. No outsourcing, don't just simply, you know, have others copy and paste their answers for you, because again, the goal is for you guys to learn. We're not out here to get you. We're here to help guide you and teach you. All right, so any questions on any of the administrative aspects of the course?
I have a question about the problem set. So the graduate version includes additional problem on current research. What current means was only research or something?
Okay, Ko, you're gonna have to repeat that question, sorry.
So it says for the graduate version, there's an additional question on the problem sets that relates to current research. Can you explain that a little more?
The P-sets are actually already posted, so you can actually take a look at what these questions entail. So it's sometimes it's about a recent paper, and it will basically tell you, you know, do you agree with that particular approach? Or, hey, how would you do this better? Sometimes it's a more theoretical aspect of an existing algorithm that we see in the other part. So it varies by P-set, but yeah, I guess we can provide a little more information about that, and also, you know, add it to the slides, but also, you know, it's already on the Stellar website, at least the pieces from last year. Other questions?
Okay.
Okay, great. So I always ask the students, why computational biology? And I got a lot of answers as to why you are interested in computational biology. But my question here is, why do we even study computational biology? What makes computational biology so special? It's going to be a little tricky to do this over the web link, but let's see if you can provide some answers as to why computational biology. And then Koi, maybe you're going to be repeating each of them.
Okay. Why is computational biology important? Anybody? Yes.
Because in modern biology, there's a lot of data and a lot of information. You gotta need something like that.
Okay. Modern biology has a lot of data, and computer science can help us process that.
Good. That's usually answer number one, lots of data.
Yes.
Awesome. Notice there's a lot of room for additional bullet points.
Yes.
Bioengineering is currently sort of wishy-washy and hand-wavy in computational biology. That's to get better designs, and because lab work takes so much time, fewer iterations.
It can help us optimize.
So guiding experiments to make them more efficient. Very, very good. I mean, that's changing a little bit in the sense that right now we can do massive experimentation in parallel. So in a way, it becomes a little easier to do many, many experiments and then figure out what the heck does the data mean. But absolutely, for, you know, knowing what gene to perturb, if you're trying to dissect the disease mechanism, you know, there's no such thing, there's no better thing than just being able to say, hey, I think this disease is acting in this cell type and affecting these particular genes, so that's how you should guide your experiments, because otherwise the search space is just huge. Other answers?
Yes.
Understanding the biological system better, because some experiments make it much.
If you speak up just a teeny little bit more, I'll be able to hear them directly. Understanding a biological system better?
Then, like you could with the wet lab experiment, because you can build a model and then inspect the model.
Very cool. So basically bringing order to biology, basically building models and sort of testing these models. Yeah, very good. Next. Is anybody speaking? I can't hear anything.
No. No. Do you have any more ideas, Manolis?
I have a few more.
I think modern techniques, sensitivity and capacity for volumetric increase so much that you need a commensurate amount of processing power in the data analysis to decompute all data.
Yeah, so one of them is compute power. Is that what you're saying? Basically, humans, even if they had all the brain power in the world, they wouldn't be able to sort of have the time to go through and, you know, shift through all the data. Is that what you're saying?
More or less.
More or less.
More or less.
Yes. Sure, yes.
It can be difficult to test things in isolation, so computational biology sort of lets us sort, you know, signal from noise.
Very cool. So basically one of them is signal from noise, and then the other one is learning about combinations. So basically on one hand, there's just so many genes. On the other hand, sometimes genes act in combinations, not in isolation. So being able to not only search through the order n super large data set, but order n squared, n cubed, and so on and so forth. Yeah, I agree. Other comments? What makes computational biology so different than, say, computational geology or physics or, I don't know, other fields? There's something that makes biology so special and so amenable to computation.
We have one person who has an answer. All right, go ahead.
Impacts people's life directly.
Affects people's lives directly.
That's good. So basically, in terms of impact, it can be very large in the very near future. Correct. So that makes it extremely important.
Lending communication to biology also allows you to store data more efficiently instead of just a stack of papers.
Store data more efficiently? What do you mean?
Like you just store it to hard disk with a bunch of numbers, ones and zeros, but like if it's paper data like decades ago, you're just a stack of paper books. And you can search through it so easily.
Koi, am I missing some keywords here?
So I guess.
It's like searching.
Well, but how is that different than physics, computational physics? I think the question. Yeah. What makes biology different than other types of computational fields? Okay.
I mean, all these answers are good. So let's see some of the ones we've heard in the past. Yeah, there are rules, so we heard that. So pattern finding, so basically the fact that there are repeating things that we can find over and over again. All about data. Ability to visualize, that's a very important aspect. Run simulations, understand temporal relationships. We talked about sort of this ability to generate hypotheses for testing rather than just searching, you know, a huge search space. We again heard that this year, proposing mechanisms and theory to explain observations. Combinations of variables, we heard that already. Efficiency, reducing experimental space to cover, talked about that. Yeah, there was some talk about infrastructure, so ability to compute rapidly and also to combine data sets. Find higher order relationships and correlations. And then the concept of basically cycling from hypothesis generation to testing, which can be condensed. And then the last one I was hoping to hear is that life itself is digital. So it's not just geology or physics or, I don't know, many other of the natural sciences. Life itself is not, you know, some weird unordered thing. You know, there's a core, there's a computational core in every single cell out there, and that's written in a digital form. Basically, humans did not invent the first digital computers. Humans are the descendants of the first digital computer. So, you know, digital computers inhabit every one of our cells. Every one of the cells reads a digital code and makes the decisions using, you know, a very well-defined cellular instruction set written in a language that we have the ability to track through computation. So, you know, the reason why I think computational biology is such a special field is because we are actually using computation to understand something that's at the root computational. And you know, that I think is a very special kind of relationship that basically we have evolved to a point that we can actually look inside our own cells and figure out the code that allowed us to make it so far and to, you know, understand. So basically, next to neuroscience, I don't think there's any other science that's as introspective as computational biology. Any other comments, having heard all of that? Any other things that you feel are missing from this list? Cool.
One more question.
Oh, one more.
Yeah, I think another powerful thing about computational biology is it can like help like experimentalists to generate like new hypotheses. Like for example, for like cancer genomics, like we can like know like what genes are affected in like patient sample. We can like actually test them in experimental systems. So I feel like that's like a connection to like web stuff.
Yeah, absolutely, completely agree. Any other comments? All right, so we're here in a very special moment in time when we can actually do that, when we can actually, you know, look inside the cells and figure out how things work. So signal to noise was one of the comments, and this is actually a little snippet of the yeast genome. So within this segment, there are 2 genes. Can you guys figure out where they are? It's easy. They start with an ATG. Here's one, and here's another one. So ATG codes for a start codon, and then you have another codon, and then they all stop with TAA, TAG, or TGA. Come on, you could have all done that. So these are the genes, and these are the control elements. So I mean, this is actual data. I'm not joking. Like, you're actually looking at a genome, and these are all the letters that actually make a yeast cell work. I mean, you know, this is 2 of about 6,000 genes that make a yeast cell work. In fact, these 2 genes are extremely important. They actually help metabolize a sugar called galactose, which is one of the primary sources of food for yeast, along with glucose. And in fact, there's motifs here that turn off the galactose processing. CCCA actually turns off galactose processing genes when you have glucose, because glucose is a more efficient sugar. And these motifs here, CGG, CCG, which are palindromes, so CGG on this strand is the same as CGG on the other strand, written here backwards and reverse complemented. So there's a motif here that's palindromic. 11 nucleotides is 1 full turn of the double helix. So this is actually a transcription factor, a protein that binds 2 different sites that are spaced by 1 turn of the helix apart from each other. And then they're right next to another one of those and another one of those, which is kind of cool. It basically shows that there's 3 different binding sites for the same factor. And this is actually the factor that comes and turns on the galactose processing gene. So GAL4 actually binds these motifs. And then TATA is the place where the TATA binding protein binds, TBP, which actually helps turn on the basic machinery for transcription and ultimately, you know, production of the mRNA molecule and so on and so forth. It's kind of cool, right, to be able to just read biology to sort of, you know, see where things are. But I'm kind of telling you the answer. If I didn't tell you that these are where the genes are, I don't know if you would be able to find them. And especially for the motifs, I don't know if you would be able to recognize those. But that's what's so special about genomes, the fact that within the same string of DNA, you have both the data that encodes proteins and the circuitry that decides when these proteins will be generated and how. So you basically have both, you know, the protein coding part, which is what a lot of people are, you know, focused on in the literature. Every time we say, hey, where's the gene for this and the gene for that? And then you have all the control elements. After about a decade of human genetics, what we realized is that in 93% of cases, common genetic variants that perturb biological function do not hit the genes directly. in 93% of cases, they actually hit the control regions. So this is not what anybody expected. Everybody was like, hey, we're going to find the gene for diabetes and the gene for Alzheimer's. Turns out that it's not just a single gene, it's like thousands of genes. And it turns out that it's not genes at all. It's actually control elements surrounding these genes. So being able to understand the genome systematically is the only way to actually understand human disease. It's the only way forward to translate thousands of genetic studies into mechanistic insights and ultimately therapeutics. So that's what we're going to be trying to do in this term, basically learning how to read and understand genomes and developing the computational infrastructure for doing that. All right, going back to the signal to noise, I told you to find these genes. I waited a full 2 seconds and you guys did not raise your hands. So let's see why. If I look for all the ATGs, they're kind of everywhere. You guys even remember where the genes are that I showed you in the previous slide? So part of what we're going to have to do is figure out how to recognize true signal from noise. How to basically figure out where are the ATGs that actually start a gene and where are the ones that are just bogus. Again, here they are. But if you didn't know that, you'd be seeing genes all over the place. In fact, you know, some people do. So basically, part of the challenge is actually extracting signal from noise. And here's sort of beyond what I showed you just now, which was 1 gene and a half, and then, you know, a few motifs. Here's the components of genomes. Here's the components of gene regulation. Here's the building parts. On one hand, of course, there's the genome, there's the genes, and then there's the control regions. So what I showed you was individual regulatory motifs, but these motifs are organized together into enhancer regions, which are distal control regions that control genes sometimes from 1 million nucleotides away, and proximal promoter regions that guide the transcriptional machinery as to where it should start transcribing. So these are the regions of regulation. And within these regions are the actual words. These are the regulatory motifs, the high resolution accessibility maps that I showed you on the previous slide. And then the regulators are the controllers that bind these regions that sort of are encoded from elsewhere in the genome, translated into proteins, and then come back into the nucleus to bind DNA. And these include chromatin modifiers and transcription factors and nucleosomes, and these are all made out of proteins that basically control sort of how the genome gets turned on and off. We're also going to be studying the dynamics, so basically how these maps are changing between cell types and across development. The networks that are built from these regulators to the distal enhancers that they control, to the target genes that these enhancers are linked to. And then the grammars, the combinations of motifs that give rise to these elements. We're going to be studying human variation, human diversity in population genomics, and how these elements actually vary from person to person and how that impacts their regulatory machinery. And also how these elements evolve, how they change across time. Turns out that different classes of elements evolve in very different ways, which allow us to recognize evolutionary signatures for different classes of elements based on the type of evolutionary pattern that they show. I'm actually losing my voice. I'm going to go get some water. Koi, can you please lead a stretch break where everybody has to actually stand up and stretch up and turn around and turn left and turn right?
Yeah, I'm actually going to talk about office hours real quick.
All right, perfect. But have people stand up while you do that.
You can stand up if you want to, while I talk about office hours. So since assignments are due on Tuesday night, and we sent out a doodle before class, and it seems that Tuesday was the best day for a lot of people. But unfortunately, I don't think that 6 to 8 will work super well for me. So I was planning to change the time to about like 5 to 6:30 on Tuesdays. Is there anyone for whom that doesn't work anymore? Okay, and then yeah. Okay. Oh, okay. Oh, I see. Okay. So right. Right, yeah, so I can do one outside of it. So Monday was the sort of flexible one for people who can't make the Tuesday time. So Would you like to make it earlier? Is that okay? Okay, yeah, we can try to arrange a time. But yeah, seems that so for most people on Tuesday, between 5 to 6 will work. All right. Yeah, I'll send out an email about it later, but I just wanted to check in.
Okay, perfect. Can you guys still hear me? Can you see my slides? Okay, awesome. So this is what we're going to be learning about. And then lastly, you know, beyond evolution, how these are changing disease, how we can start interpreting the personal genome and the personal epigenome of every person, and how these ultimately lead to variation between individuals and different phenotypes. So we're going to be basically covering each of these topics throughout the term, and then, you know, every one of them in terms of biological questions is going to be coupled with a foundational computational tool. So basically, we're going to be talking about sequence alignment and evolution, and dynamic programming, database search and hashing, how to model biological signals and hidden Markov models, how to model transcriptomes, and then clustering and expectation optimization, how to model regulatory networks with graph algorithms and spectral analysis, how to model regulatory motifs with information theory, Gibbs sampling, and again, EM, epigenomics with classification and modeling, population genetics, and then gene trees and species trees. So here's the 5 modules that we're going to be dealing. So basically, we're going to be going through the different challenges of computational biology, going from genome assembly all the way to annotation of genome, annotation of the motifs, comparison of different genomes, understanding how the genomes evolve, understanding how to analyze transcripts, how to build clusters of genes that are co-expressed, how to search for motifs within these clusters, how to build networks from the regulators to the motifs to the target genes, and what are the emerging network properties. And you know, basically the different modules are going to be going through these topics. So basically, you know, in module 1, we're going to be learning about alignment. In module 2, about clustering and classification. In module 3, about epigenomics and gene regulation, about regulatory motifs, about how to discover these motifs. We're going to be learning about epigenomics and combinations of histomodification maps, learning about evolution and phylogenetics, learning about disease and genetic association studies, basically understanding how to uncover individual nucleotides that are associated with complex traits. And once we have recovered the nucleotides, understanding how we can translate that into mechanistic insights. Learning about evolutionary signatures and how they're guided by the structure of these elements, how we can use those to recognize protein coding genes or RNAs or regulatory motifs. We're going to be learning about phylogenetics and how we can in fact represent the pairwise distance matrix of different species or different genes from each other, embedded into a tree structure, and the duality between the two. Algorithms for dealing with trees, so nested algorithms that walk up and down the tree phylogeny. We're going to be learning about coalescence and how individuals from a particular species are in fact related to each other, sometimes through the same species, sometimes across different species boundaries. Learning about gene flow between Neanderthals and humans, learning about you know, all of these different aspects. So before we go on, because we're going to be assuming a lot of biological knowledge that hopefully you saw in 7012, and for those of you in 6.7, in 728 and 760, I'd like to at least introduce some of the notions of what we should know about in this class. So this is like a crash course in molecular biology. If you haven't taken 7012, I highly encourage you looking at the course online, and maybe even attending the lectures in conjunction with class. But hopefully by now everybody has heard about the central dogma of DNA makes RNA, makes protein. This is the central dogma of molecular biology. And a lot of the course will be walking through the different layers of that central dogma. So basically starting with DNA, this is what I like to call the most noble molecule of our time. It is, absolutely beautiful in its structure and completely counterintuitive, in the sense that when Watson and Crick first discovered that structure, stealing a little bit of the work of Rosalind Franklin and others, they basically were extremely surprised that the center of the structure was in fact held together by weak hydrogen bonds, when in fact a very strong phosphate backbone was on the outside of the structure. People were initially looking for a helix that was reversed, where the core of the helix will be held together very strongly by this phosphate backbone, and then the hydrogen bonds will be outside somehow. But what Watson and Crick realized is that as soon as you put the hydrogen bonds inside, that immediately implies that this molecule can be opened and closed very easily. And that's the whole point. There are millions of these very weak bonds that actually hold the molecule together very strongly. But any one of these bonds can be undone and redone in order to actually read the genetic information that's held inside. So to me, this is just such a beautiful concept, the fact that all of life and the transmission of life relies on these very, very weak bonds. And the whole point of it is that it is actually a book that's easy to open. You just open it, make an RNA, close it again, open it, make a copy, close it again, and so on and so forth. So again, the self-complementarity sets the molecular base of heredity. If I know that I have an A in this strand, I immediately will be able to copy a T on the other strand. If I have a T on this strand, I will immediately be able to copy it into an A on the other strand. So the self-complementarity of DNA basically says that if I only have one copy of one strand, I can immediately infer the other strand. And that led to this very famous sentence that says, "It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material," which was part of their initial publication. But they basically said, you know what, let's write a short paper rather than try to explain all of this at great length. And this little sentence basically says, yeah, we get it. We're going to publish a longer paper about that. Basically, knowing one strand creates a template for the other. So what are these chemicals? Again, I mentioned these weak hydrogen bonds. C and G base pairs have 3 hydrogen bonds. They're called strong bases because they're held together very strongly, 50% more strongly than A and T bases that only share 2 hydrogens. The phosphate backbone that we talked about earlier runs here from the 5' atom to the 3' atom of these rings on the outside. So that's why we call it the 5' to the 3' direction of reading DNA. And you'll notice that the 2 strands are not just parallel, they're anti-parallel. So this one runs from the 5' to the 3' direction this way, and the other one from the 5' to the 3' direction the other way. Previously, I mentioned that CGG in one strand is equivalent to PCG in the other strand, and vice versa. So that basically means that, every time I have to reverse complement something, I have to, read it in one direction here, and then the same thing will be in the same direction there. Again, the bases are hidden on the inside, the phosphate backbone on the outside, and then this allows low energy opening and reclosing of the 2 strands. And the only possible pairings are A with T and T with G. Based on this, we can abbreviate A and T bases as W, S or T, C or G bases as strong, and then these 2 bases have an amino group, and then these 2 bases have a keto group, and these 2 bases are purine, and these 2 bases are pyrimidine. So if we want to abbreviate pairs of nucleotides, we can use R, Y, W, S, M, and K to basically refer to any possible combination of two bases. So that's all about DNA. And then surrounding DNA is, of course, a modulation of that primary sequence with the epigenome. So prokaryotic cells have basically DNA floating around. Eukaryotic cells have DNA nicely packaged inside the genome, inside the nucleus. And this packaging happens through many, many different layers. And that's where epigenomics sits. The DNA is wrapped around nucleosomes. There's 147 base pairs of DNA around every one of these nucleosomes. And these nucleosomes are packaged together in higher order structures that create loops. We've learned a tremendous amount about that in recent years, and we're going to have a lecture on the 3D genome and how this is packaging is possible, and ultimately into these chromosomes. And this packaging is not just structural, it's also informational. In addition to the information encoded in the ACGT of the DNA code, you have additional information encoding in the packaging of the DNA. So methylation marks can occur in Cs of CpG dinucleotides, which indicate that this region should be repressed when it's a regulatory region. And that's one form of epigenetic modification. Another one is on the accessibility of DNA, whether a particular protein can actually go in and bind the DNA, or whether that region is actually inaccessible because it's wrapped around these nucleosomes. And then lastly, I mentioned how 145 base pairs of DNA are wrapped around these proteins. So these are histone proteins. There are exactly 8 histone proteins surrounding making up every one of these nucleosomes. And every one of these 8 histone proteins has a long tail of amino acids sticking out, which can be post-translationally modified. This can be acetylated, this can be methylated, this can be ubiquitinated, phosphorylated. And these modifications actually carry a lot of meaning. So they can actually tell the genome that this particular region encodes an enhancer, for example, based on the specific combination of histone modification marks, even without looking at the DNA, which allows very rapid lookup of specific regions that might be important in different cell types. Any questions so far? Again, we're going to have a whole lecture on every one of these topics, so don't panic if you can't, make up all the keywords that I'm using right now. So this is a close-up of these nucleosomes. So you can see here the 8 histone proteins and DNA wrapped around twice around each nucleosome, and then the long amino acid tails, and then the modifications on each of these tails. So we're going to be talking about H3K4 trimethylation. So that's histone 3. Of all of these different types of histones, there's histone H3, H4, H2A, H2B, which are the most common ones. Lysine 4 basically means that there's 1, 2, 3, 4 residues. The fourth one is a lysine, and it has 3 methyl groups, or a single acetyl group, and so on and so forth. And then we're going to be looking at this information across different organs and across different tissues and across different cell types. We're going to be able to map this epigenomic information, which is extremely variable across all of these different modalities. And then learning about sort of how to interpret epigenomic information by combining accessibility, DNA methylation, and histone modifications to basically infer this histone code, basically. So that's all about DNA, and then epigenomics surrounding DNA. Going forward to RNA, we're going to be looking at how genes are transcribed. So DNA is transcribed into RNA. We're going to be looking at the regulation of transcription and sort of how genes are controlled and how RNA is made, and ultimately how these are translated into protein. The proteins are the workhorses of the cell. They are the ones that play the most diverse kinds of functions. They hold the cell together. They build the cytoskeleton. They control movement of particles and metabolites across the cell. They have extremely diverse structures in 3 dimensions. This is one of these structures, and you can see here DNA in the background, and then here's a protein with 1 helix, a turn, another helix, a turn, another helix. So these helices are in fact contacting the bases of the DNA and recognizing these bases through the chemical properties of A, C, G, and T. Without opening the DNA to read these bases, they can actually feel from the side that, oh, this is an A base because it has this atom here. And that's how a lot of regulators are in fact finding their targets, by directly contacting these bases from the side without opening the DNA helix, and then recruiting other machinery. So amino acids are much more complex than nucleic acids. So nucleic acids are kind of boring. There's only 4 of them, you know, without counting U. And amino acids are much more complex. So people used to think that proteins must be the building blocks of life because there's just so much information that can be encoded. And this is possible through this, you know, amino acid backbone, an amino end and an acidic end, COO minus, a carboxyl end, and then a variable side chain. And these are sort of all of the different side chains and how they can encode this extreme diversity in protein functions. You pair those together, and then that's how you make a protein. And then you translate RNA to protein by incorporating these adapter molecules known as tRNAs that basically will recognize 3 nucleotides at a time, bring in the corresponding amino acid, and then link it together inside the ribosome. This is the genetic code. You can basically read it by looking at the first codon position, second codon position, and third codon position. So CCC, for example, encodes proline. But it turns out that CCA, CCG, and CCU all encode proline. So that actually leads to a very clear evolutionary signature of protein coding genes, where the third codon position can actually be much more variable than the first 2 codon positions because it doesn't actually affect the protein translation. So through this genetic code, we basically end up with a start codon that's also always a methionine. We saw that in the ATG of the first slide that I showed you with DNA on it. And then the 3 different stop codons, TAG, TGA, and TAA. So, of course, once proteins are made, the loop closes because proteins, in fact, control both RNA and DNA. So this is what leads to this huge diversity of functions and this complexity of gene regulation. We have recognized year after year just how incredibly complex RNAs are. We like to think of them as sort of this middle layer where DNA makes RNA makes protein as just a messenger on the way to protein. But in fact, RNA itself can be extremely complex in its functions. So there's many small RNA pathways, microRNAs, piRNAs, SNRNAs, you know, 21U RNAs. And there's many long non-coding RNAs that are recently recognized, which are thought to play scaffolds for protein TF binding and for 3D structural assembly of RNA. The basic building blocks of gene regulation is that there are these small nucleotide motifs between 6 and 8 base pairs long that are recognized by transcription factors. These bind in complex assemblies that then lead to recruitment of machinery that can rearrange the epigenome, open up a region much more stably, and ultimately recruit RNA polymers that will make RNA and ultimately protein. So we can actually recognize the motifs associated with each of those and find patterns in the DNA that are repeated in regions that are coordinately active. And it turns out these patterns correspond exactly to the recognition sequences of corresponding motifs. So we can use those to start recognizing what are the connectivity patterns of regulators, target genes, the proteins they produce, and other regulators, to basically recognize functional modules that are repeatedly used throughout cellular functions. And we'll also talk about techniques for perturbing these motifs in massively parallel ways. So you can actually perturb thousands of these motifs in a single experiment, basically understand how they're affecting function of the corresponding regions. We can recognize the emerging networks of connectivity and the modules and the network motifs that emerge from that, and then start designing circuits for utilizing these principles to then go and edit cellular functions. In one paper that we published a few years earlier, we were able to transform one organ of the fly into another organ of the fly by changing a single microRNA, exactly as was predicted by our regulatory patterns. And then we were able to sort of construct these mutant flies and watch organs change right in front of our eyes. And this was by recognizing this basic machinery of XOR loop that basically only allows one or the other of these two processes to function, but not both. And as soon as one of them is turned on, it sort of very strongly inhibits the other one. So, of course, DNA is not immutable. Otherwise, evolution wouldn't even be happening. We would just be still a bunch of bacteria floating in ponds rather than, incredibly complex life forms that, I don't know, make computers and travel to the moon. And this is all possible because we're actually kind of bad at copying DNA. We make mistakes sometimes. And these mistakes are the basis of how evolution works, but they're also the basis for how disease occurs. And one particular class of mutations are single nucleotide polymorphisms, or SNPs, that are extremely frequent in the human genome and across the human population. So back in Africa, we were only a group of about 10,000 people that went on to populate the rest of the planet. And we had a lot of common variants that are still segregating in the human population today. And some of those common variants are at 40% frequency in the human population. And even though they're at 40% frequency, they are sometimes disease associated. So what we can do is systematically ask, what are the nucleotides that are associated in every region of the genome with specific differences between individuals? So we can take, let's say, 100,000 individuals with schizophrenia and 100,000 individuals without schizophrenia, and then look for patterns of differences in their genome that are associated with these phenotypic differences. And that's the basis of genome-wide association studies that basically look for SNPs, or single nucleotide polymorphisms, whose identity between, say, an A and a G is very strongly correlated with specific traits. So doing this across the entire genome basically gives you these kinds of plots, which we call Manhattan plots. Just like the tall buildings of Manhattan are sort of skyscrapers that are sometimes much, much taller than anything else around them. This is the same thing that you find when we carry out the same chi-square statistical test across every nucleotide that is variable in the human genome. So these now tell you that these genes here are very strongly associated with a particular disease because nucleotides near them are, in fact, underlying this. So the power of these genome-wide association studies is that they're completely unbiased. They can reveal new disease genes in locations that were previously completely unexpected to act in these processes, based on these differences between the phenotypes of individuals that carry the A allele and individuals that carry the G allele in their predisposition to disease. We can basically infer that this particular region plays a role in the disease. So going, however, from these regions to the actual function of how these regions are acting can be extremely challenging. And this is one of the topics that we're going to be covering this term, as to how we can, in fact, translate a single nucleotide polymorphism into a complex circuit alteration that leads to disease. And this is actually a picture from one of the papers that we published recently in the New England Journal of Medicine, basically looking at a single nucleotide polymorphism associated with obesity and recognizing that this polymorphism, this single nucleotide difference, changes the binding site for a protein, ARID5B, that binds normally this motif and then turns on 2 different genes that are sitting 1.2 million nucleotides away and 600,000 nucleotides away, which then turn out to be master regulators of a process known as thermogenesis, or generation of heat, that allows us to actually burn calories instead of storing them as fat. And understanding that circuitry allowed us to then go in and perturb these circuits to change the metabolism of primary cells. We were able to switch fat storing cells into fat burning cells by changing a single nucleotide back and forth. And when we went to mice, we were able to change the expression of a single gene, and then the mice lost 50% of their body weight. And that all comes from the ability to understand genomes systematically, be able to infer these circuits, and then be able to go and manipulate these circuits. And then the last topic we're going to be looking at is genome evolution, how things are changing across time. So this is the goal for the term. This single page here will summarize a lot of what we're going to be going through. I focused a little bit both on the topics that we're going to be covering, the modules that we're going to be going through, the foundations and frontiers within each of the modules, the P sets, and the project guidelines. So any questions about the class or the topics or the organization? Who's excited to learn a ton of information about computational biology? Raise your hands. Awesome. Good. Me too. So I look forward to seeing everyone next week, and then we will start with alignment, dynamic programming, and then database search next week. So don't forget to sign in Friday as early as next week, and then we'll take it from there. Thanks a lot, Corey. Thank you, everyone. Thanks for putting this together. Bye-bye.